Hi, I'm Stephen Silvestrak, and for the next few minutes we're going to explore several of the important functions that cellular proteins fulfill. When you think about proteins, one of the first thoughts conjured up, especially in the biochemistry section, are the many different enzymes that keep our bodies humming along. You might also think about peptide hormones that help with systemic signaling. But our bodies use amino acids for way more than triggering G-protein coupled receptors or lowering activation energy of life-sustaining reactions. And the MCAT believes it's important that you are familiar with these other non-enzymatic functions. So for the next few minutes, we'll examine the essential structural, motor, binding, adhesion, and immune functions that cellular proteins can fulfill. Our goal is to be able to identify several non-enzymatic proteins and their functions so that we can answer MCAT questions that specifically address which proteins are involved in which functions, like this question, which asks about the protein components of epithelial tight junctions. Structural proteins use their characteristic shape to support the cell. A common thread between all structural proteins is the presence of repeating secondary structures. Secondary structures, like an alpha helix or a beta-pleated sheet, can stand alone or organize together. When there are multiple secondary structures organized together, we call it a motif. Depicted in this molecule is a simple motif of two beta sheets, connected by a few amino acid residues making up a random coil. As the beta sheets interact with each other, those intermolecular forces will maintain a strong tertiary structure that give each structural protein its characteristic shape. Collagen is a strong, yet flexible extracellular structural protein comprised of three intertwined alpha helix motifs. You can think of it as three alpha helix motifs braided together for an improved strength. Collagen is a major player in our connective tissue, and it is found in the extracellular matrix in everything from skin to cartilage, muscle to bone, and even our eyes. Another structural protein found in the connective tissue is elastin. A key feature of elastin is that it is highly coiled and cross-linked, giving it the ability to stretch and return to its original shape. These coiled segments, shown in dark blue here, can pull apart and then will also recoil to their original shape. Keratin makes up the intermediate filaments of epithelial cells, meaning that they are essentially the cellular skeleton. In the image on the right, you can see the picture of a cell with a special fluorescent stain that binds to keratin, and in this case makes the intermediate filaments glow in a hauntingly beautiful yellow. It is important to contrast the function of keratin with the function of the collagen and elastin we just discussed. Whereas proteins in the extracellular matrix of connective tissue hold together the entire tissues from the outside of the cell, intermediate filaments give the shape and support to each individual cell from within that cell. Keratin helps give each cell its shape, keep the nucleus in place, and also help cells stick together. Keratin takes on many structural functions, and you may also recognize it from its role in waterproofing skin and forming hair, fingernails, and toenails. Actin, shown here with a different fluorescent stain, is another structural protein that you probably recognize as a major component in muscles. And it does compose both microfilaments, which are the thinnest filaments of the cytoskeleton, and myofibrils, or muscle fibers, the basic rod component, aka thin filament of a muscle. Actin is the most abundant structural protein in eukaryotic cells, and in addition to its protective role, actin is also polarized. Because there is an ordered separation of positive and negative parts of actin, when actin is paired with a motor protein, the motor protein will move in one direction along the actin, allowing for coordinated movement of the cell. Tubulin, fluoresced here in blue, is similar to actin in that it is polarized and serves as a structure on which a motor protein can move in a predictable direction. Tubulin is most famous for its role in providing the framework for separation of chromosomes during mitosis and meiosis. But tubulin is also the framework for intercellular transport between kinesin and dynein, two motor proteins we'll explore in a little bit. Also, keep in mind this key differentiator for the MCAT. The positive end of tubulin is away from the nucleus, and the negative end is toward the nucleus. Both of these last two structural proteins, actin and tubulin, serve as the structural framework on which motor proteins can move. Myosin moves along actin, and you probably recognize it as the thick filament from a striated muscle fiber sarcomere. Pictured here is the myosin head, and collections of these myosin fibers bind together as the thick filaments in a sarcomere. By hydrolyzing ATP, these heads can move along the actin myofibril, and as the myosin head moves, the sarcomere will contract. Although myosin moves along actin, contracting the cell itself, two other motor proteins, kinesin and dynein, are responsible for moving things within the cell. Kinesin and dynein have two ATPase domains, shown here in blue and green, that hydrolyze ATP sequentially, allowing the motor protein to walk along the microtubule in a rotation motion. The two legs of the motor protein walk in a three-part cycle. To start, one leg is attached to the tubulin dimer, shown here in blue, and the other leg, shown here in green, is free but has an attached ADP. When ATP affixes to the attached leg, the motor protein can rotate. Then, the attached ADP will dissociate, allowing both legs of the motor protein to latch onto the tubulin. When the attached ATP is hydrolyzed and the phosphate leaves, the leg that used to have the ATP can now detach from the tubulin, and the motor protein is ready to start this three-stage cycle over again. The next step in the walk would be an ATP attaching to the green leg, resulting in the rotation of the motor protein. Both kinesin and dynein hop along the beta subunits in this way, but they travel in opposite directions. Dynein moves in toward the nucleus, and kinesin moves out from the nucleus. A quick way to remember the difference on test day is to think about dyning in with dynein and carrying out with kinesin. The third type of cellular protein we're going to explore are binding proteins. Although there are many types of proteins that bind to molecules, like calmodulin or DNA transcription factors, hemoglobin, which is shown here, is an absolute MCAT favorite. You are probably familiar with the basics of the 4-subunit hemoglobin structure, where each iron or heme containing subunit can cooperatively bind one O2 molecule. Although binding proteins attach themselves to molecules, there is a whole other class of proteins that help cells attach to each other. These are cellular adhesion molecules. Cadherins participate in the calcium-dependent adhesion between cells. Cadherins are typically found in intercellular junctions, where they attach to other cadherins through the extracellular space and join two adjacent cell membranes. Although the cadherin system works well for cells that are naturally stuck to their neighbor, a different system is used to help cells that aren't always next to each other. For example, as blood flows by the endothelium, the neutrophils in the blood will, in order to fulfill their function, need to drop out of the bloodstream by adhering to the endothelial lining. Integrins, which are integrated into the cell membrane of one cell and bind to the extracellular matrix of another, help in this process. Selectins are similar to integrins, except selectins selectively bind to carbohydrates on another cell. Both selectins and integrins regulate how neutrophils can leave the bloodstream and enter the surrounding tissue, a process called extravasation. Speaking of the immune system, B cells release immunoglobulins, otherwise known as antibodies. These proteins have a tertiary structure of two heavy chains and two light chains connected together by cysteine disulfide bonds. Both the light and heavy chains have constant domains and variable domains, and it is the different combinations of variable domains in the different antibodies that allow each antibody to attach to a different antigen. The part of an antigen that an immunoglobulin can connect to is called the epitope, so we call the combination of variable domains the epitope binding site. As antibodies attach to their respective epitopes, they will further activate the immune response. Let's review some of the MCAT knowledge we just covered with some MCAT questions. Which protein type would you expect as a structural junction between intestinal epithelial cells? The calcium-dependent adhesion of cadherins is responsible for joining together cell membranes all over the body, including, of course, the intestines. Selectins and integrins are cellular adhesion molecules, but they are more associated with neutrophil extravasation, and immunoglobulins, or antibodies, are involved with antigen binding and activation of the immune response. Cadherins are the only choice that involve continued, stable connections between cells. Let's answer another MCAT question. Which protein is not involved with the extravasation of neutrophils? Selectins, integrins, and glycoproteins are all involved with the binding of a neutrophil to the epithelial lining so the neutrophil can leave the bloodstream and enter the tissue. Kinesin, on the other hand, is a motor protein and not directly involved in this cellular adhesion process. Let's review some of the major MCAT points we explored. Collagen is a structural protein made of 3 intertwined alpha helices and is found in the extracellular matrix. Tubulin is another structural protein, but it provides a polarized pathway inside the cell for motor proteins like kinesin to move out and dynein to move into the nucleus by rotating along the beta subunits. The most tested binding protein on the MCAT is hemoglobin, which has 4 subunits that each cooperatively bind an O2 molecule. Cadherins are the calcium-dependent adhesion protein that form tight junctions between cells. And selectins and integrins are other adhesion proteins that are involved in neutrophil adhesion and extravasation, allowing this white blood cell to patrol the tissue for foreign invaders. Finally, immunoglobulins, or antibodies, are comprised of light chains and heavy chains, and their variable region uniquely binds a particular epitope on a specific antigen. This binding will further signal immune system activation. This concludes our examination of non-enzymatic cellular protein function for the MCAT. Please review as necessary, and as always, best of luck on test day.